A very good afternoon, dear students. I am Professor Dr. Vikas Mishra from GSMM Medical College, and in this lecture we'll be discussing about preservation of microorganisms. So what do you basically understand by preservation? To maintain pure cultures for extended periods in a viable condition, in a living condition, without any genetic change is referred to as preservation. यानी कि हमने उसे प्योर कल्चर फॉर्म में उस बैक्टीरिया को या माइक्रो ऑर्गेनिजम्स को बहुत देर के लिए हमने स्टोर करके रखा हुआ है विदाउट उस पर कोई जेनेटिक चेंज हो, कोई म्यूटेशन हो जिससे वो अपनी फिजिकल प्रॉपर्टीज को चेंज कर सके, वो वैसी स्थिति में रहे जैसा वो प्लेन उसमें रहता है. दैट प्रोसीजर इज नोन एज preservation. So what are the aims of preservation? The aim of preservation is to stop the cell division at a particular stage. That is, to stop their microbial growth or at least lower down the growth rate so that they do not multiply. Due to this, the toxic chemicals which are released during metabolism of multiplication of these microorganisms, they are not accumulated inside the recovery medium. Hence, the viability of the microorganism is not affected. So, by stopping the cell division at that particular stage, we stop the microbial growth, and due to this, there is no metabolism, and the metabolic products, the excretory products are not released into the circulatory medium. Therefore, the medium is not toxic. Because during multiplication what happens, that bacteria multiplies and this process continues. During multiplication, they release their certain metabolic products in the recovery medium, and they keep on dividing, they keep on using the nutrients which are present in that recovery medium. So the nutritive medium gets exhausted and the toxic accumulation of metabolite occurs, which is having deleterious effect on these microorganisms. So in this preservation, this thing is stopped. Then the other aim of preservation will be to avoid the contamination. That is, the microorganism is made available in pure culture form. It is not contaminated by any other environment microbes. So we get the pure culture whenever we are requiring it in recovery stage. Then we also, our aim of preservation is to restrict the genetic change, because if there is genetic change in microorganism, then mutations may occur. So the physical properties of that bacteria can change. For example, if we are talking about Escherichia coli, we all know that Escherichia coli is a lactose fermenting bacteria. It ferments lactose. So during preservation, this lactose fermenting property is not changed, is a better form of preservation. But if the bacteria multiplies, there is mutation, there is genetic change. So this property of lactose fermentation will be changed and bacteria becomes non-lactose fermenting. So during recovery, when we are inoculating it into a differential media such as Mekong ki agar, which utilizes lactose as a byproduct. So Escherichia coli in Mekong ki agar appears pink color due to lactose fermentation. But if there is any mutation or genetic change due to repeated subcultures or any mutation, that bacteria will become non-lactose fermenting. So instead of pink colonies, we'll get pale colonies. That is, the physical property of that bacteria has changed due to mutation or genetic change. Therefore, the aim of preservation is also to restrict any genetic change and mutation does not occur. So in all, what is the aim of preservation is, number one, to stop the cell division so that toxic metabolites do not accumulate which have the deleterious effect on microorganism. Number two, there is no contamination in that recovery medium and we get a pure culture form whenever we require. And lastly, there is no genetic change, no mutation which could change the physical property of that bacteria. There are certain preservation methods. The methods of preservation are mainly of two types. Number one is short-term methods and the other is the long-term method. Number one, periodic transfer to fresh medium. Number two, storage in saline suspension. Number three, storage at low temperature, that is either by refrigeration or cryopreservation. Then storage in a sterile soil medium, and preservation by overlaying culture with mineral oil. And lastly, lyophilization or freeze drying. These are the methods by which we preserve the bacteria. I will repeat once again. What are the preservation methods? Number one, periodic transfer to fresh medium. Number two, storage of that preservation bacteria in a saline suspension. Number three, storage of that bacteria in low temperature either by refrigeration or by cryopreservation. Number four, storage in sterile soil medium. Number five, preservation of that microbe by overlaying culture with mineral oil. And lastly, by lyophilization or freeze drying method. So these are the preservation methods which are routinely encountered to preserve any microorganism. The short-term method, that is periodic transfer to fresh medium. The culture can be maintained by periodically preparing fresh cultures from previous stock cultures. How is it done? The bacterial subcultures are done by using a recovery medium, and these subcultures remain viable for at least 2 weeks to 4 weeks, and for fungus for at least 3 months to 4 months. The aim is to maintain the viability. The process of subculturing is repeated. It is simple and advantageous method. So from the previous stock culture, we can periodically transfer it into a selective culture media and keep the bacteria viable for at least 2 to 4 weeks and fungus for at least 3 to 4 months by repeated subculture. So how is it done? The periodic culture is done that the culture media is prepared in a tube bottles with a screw cap in it. We first heat the inoculation group. We take the stock culture and we put it in that recovery or preservation medium. We inoculate that. After inoculation, we tightly screw cap that vial, and again we sterilize the inoculation loop for preparing the next batch. And now this subculture media in which we have subcultured, it will remain viable for at least 3 to 4 weeks, and we can utilize these bacteria for various quality control processes during the period. And whenever we require, we again subculture into a new fresh culture media. But mind you, the repeated subcultures, after 4 or 5 subcultures, there may be a chances of genetic change, that is mutation may occur, and bacteria may lose their certain properties. Therefore, it can be used for short subculturing, that is 1, 2, 3 or 4 subcultures are done. Then we can also preserve the bacteria by using a material known as glycerol. Bacteria can be frozen using 15% glycerol. The glycerol is diluted to 30%, and to it an equal amount of glycerol and any culture broth which is to be prepared, which is to be preserved, is mixed, and they are dispensed into tubes and then frozen at minus 10 degrees centigrade so that the cell division is inhibited and bacteria may remain viable for the following period. Please. So the preservation of bacteria in glycerol, that is bacteria can be frozen using 15% glycerol. How is it done? Glycerol is diluted first to 30% and then equal amount of glycerol and cultured broth is mixed together. It is dispensed into red tubes and frozen at minus 10 degrees centigrade. So at this stage the cell division ceases. Bacteria cannot divide at that moment. So if it will not divide, it will not release its toxic metabolites into the medium. It will not get contaminated. It is screw cap, and it will not, even since it is not multiplying, there won't be any genetic change. But the viability of organism in 15% of glycerol is quite variable. For example, Escherichia coli, a gram-negative lactose fermenting bacteria, and Streptococcus pneumoniae, a fastidious bacteria which is causing pneumonia and meningitis in neonates. It is a gram-positive bacteria. These two bacteria may remain viable for at least 5 months in 15% glycerol, whereas Haemophilus influenzae, a fastidious gram-negative bacteria, would remain viable only up to 4 months. Neisseria meningitidis, a gram-negative diplococci, may remain viable in 15% glycerol for up to 6 weeks, whereas the same family, Neisseria, but gonorrhea, causing female genital tract disease, which is a sexually transmitted disease known as gonorrhea. Neisseria gonorrhea may remain viable for only 3 weeks. So the viability of organism is quite varied. Normally, Enterobacteriaceae will remain viable for at least 5 months. Fastidious bacteria such as Haemophilus influenzae will remain viable for 4 months. Neisseria meningitidis for 6 weeks and gonorrhea for 3 weeks. Then storage by refrigeration. The cultured medium can be successfully stored in refrigerators or cold rooms when the temperature is maintained to 4 to 6 degrees centigrade or even 2 to 4 degrees centigrade. At this temperature, the range of metabolic activities of microbes is not completely diminished, but it is almost slowed down greatly, and only small quantity of nutrients will be utilized. Therefore, the exhaustion of nutrients in this media will be slower, but it will get exhausted after few weeks. But the multiplication will occur in that. It will be slowed down in this media due to refrigeration at 2 to 4 degrees centigrade. Therefore, this method cannot be used for very long time because these toxic products would get accumulated, which can further kill the bacteria. So if you subculture the preservating bacteria in a culture media and refrigerate the bacteria for at 2 to 4 degrees centigrade for at least 2 weeks, it is okay. But if you incubate it at 2 to 4 degrees centigrade for more than 15 days, then there may be chances that the bacteria, you cannot get back the bacteria because it is already dead. Why? Because there was slower multiplication. The growth was not completely ceased. The multiplication was not completely ceased. So they were continuously utilizing, although at slower rate, the nutrients, and they were releasing the toxic metabolites into the medium, although at slower rate. But at threshold level, more than 15 days, almost all of the nutrient is utilized, and there is toxic accumulation of metabolites which will kill that bacteria. So after 15 days, you will not get the bacteria in recovery medium. Therefore, for short preservation, you can acquire this refrigeration method, but for long preservation, this is not the method of choice. So what are the long-term methods? Long-term methods employed are mineral oil or liquid paraffin storage. In this method, sterile liquid paraffin is poured over the slant culture of microbes or even the liquid cultures and stored upright at room temperature, whereas cultures can also be maintained by covering the agar slants by sterile mineral oil, which is stored at room temperature or preferably at very low temperatures ranging from 0 to 5 degrees centigrade. Now, use of this paraffin oil or sterile mineral oil would limit the oxygen excess that would further reduce the microorganism metabolism and growth, as well as to cell drying during preservation. It will rehydrate it. So the preservation period for bacteria from genera Azobacter and Mycobacteria is roughly from 7 to 10 years, and for Bacillus it is 8 to 12 years. So this can be employed as a long-term method. Why? Because the growth of bacteria will be ceased, there will be less of contamination, there will be less of mutation, and the bacteria will remain in its original form in that particular recovery culture medium for a long span of time. That is mineral oil or liquid paraffin. But mind you, this liquid paraffin or mineral oil should be sterilized prior to preservation, and you should check for its sterility. Otherwise, it could lead to contamination of that preservation medium. Then, storage in saline suspension. Bacterial cultures may also be preserved in 1% salt concentration in screw cap tubes to prevent their evaporation. The tubes may be stored in room temperature, and whenever there is need of transfer, it is made on agar slant. How it is done? That first you prepare 1% salt concentration agar slants in screw cap bottles, and then you inoculate with a sterile loop the bacterial culture which is to be preserved, and then it is screw cap tight and it is stored at room temperature. And whenever there is need for that bacteria for recovery, we take out the inoculation and inoculate in a fresh culture media and incubate it to get the colonies back. So this is one of the method which can be used for preservation of bacteria. It is quite good method. It will decrease the level of contamination also, and it will cease the metabolism of the bacteria also. Then immersion in distilled water can also be employed. It is another inexpensive and low maintenance method for storing fungal cultures. And how it is done? That the fungal culture agar slants are immersed in that distilled water, and fungi can be stored in this method up to 20 degrees centigrade, which may survive for 2 to 10 years or even more, depending on the species of that fungus. So usually immersion in distilled water is employed for preservation of fungus for a longer duration. Then for sporulating fungi, it involves inoculating agar slants or any preferred media with fungal cultures and then incubating them at 25 degrees centigrade for several weeks to induce sporulating forms. That spore forms are released. Then sterile distilled water, that is 6 to 7 ml, is added aseptically to that culture, and the surface of culture is scraped gently with a pipette to produce a spore and mycelial slurry. This is kept in sterile glass vial at 25 degrees centigrade, and to retrieve a culture, 200 to 300 microliter of this suspension is removed from the vial and then placed on the fresh medium. So sporulating fungi, what do? We convert it from vegetative form to sporulating form, and distilled water about 6 to 7 ml is added aseptically to the culture, to the surface of culture, and then it is scraped gently with a pipette to produce a spore or mycelial slurry. This is further kept in sterile glass vial at 25 degrees centigrade to retrieve a culture. 200 to 300 microliters of this suspension is removed from the vial and placed on fresh medium. Then storage in sterile soil. This is mainly applied for the preservation of, again, sporulating microorganisms which can sporulate, such as fusarium, penicillium, alternaria, rhizopus. All these are fungus. They are proved successful for storage in sterile soil. Soil storage involves inoculation of 1 ml of spore suspension into the soil. It is autoclaved. That soil has already been autoclaved twice, and then it is cooled down, and after that inoculation of 1 ml of spore suspension is done into that soil, and then it can be incubated at room temperature for about 5 to 10 days. The initial growth period would allow the fungus to use the available moisture within the soil and gradually to become dormant. After that, the bottles may be stored in refrigerator, and the viability of organisms may be around 70 to 80 years. So particularly for fungus such as penicillium, fusarium, alternaria, rhizopus, this method of storage in sterile soil, which has been sterile by autoclaving it twice, can be employed, and we can get the viability of organism for as long as 70 to 80 years. So this method is also employed for fungal preservation. This slide shows that various fungal species has been used for the preservation in this media. Lastly, cryopreservation. First of all, we need to know what is cryopreservation. Cryopreservation is freezing in liquid nitrogen of temperature less than 196 degrees centigrade or in a gas phase above the liquid nitrogen, that is minus 150 degrees centigrade. This cryopreservation helps the survival of pure culture for long time storage. In this method, the microorganisms of culture are rapidly frozen in liquid nitrogen at 196 degrees centigrade in the presence of stabilizing agent. As we have already discussed, the stabilizing agent, one of the important stabilizing agent is glycerol, and the other is dimethyl sulfo oxide, which is known by DMSO, which will prevent the cell damage due to formation of ice crystals, and it will promote the cell survival. Now, by this method, species can remain viable for at least 10 to 30 years without undergoing change in their characteristics. So cryopreservation is freezing in liquid nitrogen at minus 196 to minus 150 degrees centigrade. These pure cultures can be stored for at least 10 to 30 years. The only disadvantage of this cryopreservation of freeze drying is that it is comparatively a very costly method, and you need to have certain specialized equipment for this, and certain technical training is also required for the lab personnel, how to perform this cryopreservation and how to recover from that cryoprecipitate the bile. So this method is usually employed in research activities and also for commercial availability, such as certain commercial companies which are preserving certain ATCC strains of bacteria which are utilized for standardization purposes are being used by this cryoprecipitation. They are being freeze dried and they are being kept. Even bacteria which is used for vaccines and production of toxin is also being employed by this method, cryoprecipitate. This is one of the best method for preservation of bacteria. The only disadvantage of this is that it is available in freeze dried form and it is quite costly and you require certain technical training. This is how you can see in this slide that in a culture tube you can preserve the bacteria in liquid nitrogen for long time. Then you can also store the bacteria in silica gel. Microbes can be stored in silica gel powders at lower temperature for about 1 to 2 years. Not very long, but 1 to 2 years you can store it in silica gel. The basic principle in this technique is quick drying or desiccation at very low temperature, which allows the cell to remain viable for a longer period of time. So some of the species which are preserved on anhydrous silica gel are such as Saccharomyces, Aspergillus, Pseudomonas, Escherichia coli, and many other gram-negative bacteria. So both fungus as well as gram-negative bacteria can be stored in silica gel by quick drying at very low temperature. Although this is a good method for storage, but not for a very long period of time. Yes, you can store it for at least 2 years in silica gel. Lyophilization or freeze drying is also a process which is being used for preservation. Freeze drying is a stabilizing process in which a substance is first frozen and then the quantity of solvent is reduced either first by sublimation, that is primary drying stage, and then by desorption, that is secondary drying stage. So first is primary drying stage by sublimation and secondary drying stage by desorption. Better preservation occurs with freeze drying than with other methods because freeze drying reduces the risk of intracellular ice crystallization that may compromise with the viability of that bacteria. So by this method you can accomplish your aim of not getting the preservation bacteria to crystallize into ice form, which will compromise with the viability of that bacteria. Removal of water from the specimen effectively prevents this damage. So it is almost freeze dried. Water is absolutely removed. So lyophilization is the greatest with gram positive bacteria, even spore formers, and decrease with gram negative bacteria. But the viability of this preservation bacteria can be maintained for as long as 30 years. So lyophilization or freeze drying is a very good stabilizing process in which the preservating bacteria will be first frozen and then the quantity of solvent will be reduced, water will be removed. So first there will be a drying stage in which there will be sublimation, and then there will be a secondary drying stage in which there will be desorption, that is removal of water, and you can acquire this preservating bacteria as long as 30 years you can store in this area. So large number of vials of dried microorganisms can be stored with limited space, and organisms can easily be transported for long distances at room temperature by this method. This process combines freezing as well as dehydration. So the organisms are initially frozen and then they are dried by lowering the atmospheric pressure by using a certain specialized mechanism such as vacuum apparatus. So the process basically has to be involved in 4 stages. Number 1 is pre-treatment, in which you prepare the sample preparation occurs. Then is freezing. Number 2 method is freezing, in which you perform solidification. Then primary drying, as I told you earlier, sublimation, and secondary drying, that is removal of water, desorption, and this is may be carried out by certain vacuum apparatus. So this cycle is being shown of freeze drying in this slide. The time taken is approximately 1 hour, as you can see in hours, and the shelf life temperature can vary from 0 degrees centigrade up to 10 degrees lower, minus 50, and above to 2 to 8 degrees centigrade. So first process is solidification, in which you decrease the temperature up to minus 40 to minus 50 degrees centigrade. Then by using a vacuum apparatus, you first convert it to primary drying by ice sublimation, in which 5 to 10% of water is still left, and the temperature is used in this primary drying is up to 2 to 8 degrees centigrade. Then you acquire slightly higher temperature, that is 8 to 10 degrees centigrade, in which you perform secondary drying, in which desorption or removal of water occurs. So 5 to 10% of water is almost converted to 1% of water. Okay. So at the freezing point, when you first freed, you solidified it, there was availability of water was 95 to 90%, which was converted into 5 to 10% by primary drying, that is ice sublimation, and of course lastly by secondary drying, by desorption, that 95 to 99% of water which was available by the end of solidification is converted to only 1% during desorption. Then a word about storage vials. Glass vials are generally used for all freeze-dried specimens. When freeze-drying is performed in a chamber, double glass vial, usually screw capped, are used. Then there are certain cryoprotective agents. Two most commonly used agents are either skim milk for chamber lyophilization or sucrose for manifold lyophilization. These two cryoprotective agents can also be used for this purpose. So there are various methods for lyophilization. Number one is chamber method. In this, an outer soft glass vial is added for protection and preservation of dehydrated specimen. The silica gel granules are placed into the bottom of outer tube or vial before the inner vial is inserted, and then it is cushioned with cotton. So in chamber method, you use two glass vials. Outer soft glass vial is added for protection and preservation of dehydrated specimen, and then silica gel granules are placed to the bottom of outer vial, and inner vial is inserted above the cushioned cotton. Then there is a manifold method. It is a single glass vial method used. The storage vial must be sealed to maintain the vacuum and the dry atmospheric condition. In this, you require only a single glass vial, but you have to remember, for maintaining the vacuum, you have to seal the opening of that storage vial. So what are the advantages? There is removal of water at lower temperature, thermolabile materials can be dried, sterility can be maintained, and reconstitution during recovery is very easy. But there are certain disadvantages also. Like, number one, many biological molecules are damaged by the stress which is associated with freezing, freeze drying, or both. Example, the process of drying causes extensive damage to molds, protozoa, and most of the viruses. Hence, these microorganisms cannot be stored by this method. Therefore, the product is prone to oxidation due to high porosity and a large surface area. Therefore, the product should be packed in a vacuum using inert gas. Okay, either using a vacuum or using an inert gas, you should seal it. Then, of course, cost may also be an issue. As I told you, it is a costly process. It requires technical training for that. So you know that it is a costly process for this. Although this is one of the best method which is being employed for storage of microorganisms. So in this lecture, I have covered all of these storage examples, storage methods, out of which lyophilization is the best, but the major disadvantage is it is costly, and for certain bacteria and certain molds it may be harmful also. Although you can preserve it for as long as 10 to 30 years. So thank you very much, students, for your kind attention. If you have any doubt, you can ask them.